Hello, welcome to Watch Creative Proteomics video. Today, we are going to learn some basic knowledge about glycan microarrays. Glycans are defined as the compounds that consist of a large number of monosaccharides linked glycosidically. Glycans have diverse biological functions, and it can be classified into two broad categories. One is the structural and modulatory properties of glycans, and the other is the specific recognition of glycans by other molecules, most commonly, glycan-binding proteins. The glycan-binding proteins can be divided into two types: intrinsic glycan-binding proteins and extrinsic glycan-binding proteins. The intrinsic glycan-binding proteins recognize glycans from the same organism and mediate cell-and-cell interactions, or recognize extracellular molecules. The extrinsic glycan-binding proteins recognize glycans from different organisms, and some also mediate symbiotic relationships. They contain pathogenic microbial adhesins, agglutinin, or toxins. Due to the complexity of glycan molecules, glycomics has its own characteristics. First, glycans have a high degree of structural specificity. Second, since the synthesis of glycans is not guided by the template, it is formed under the action of various forms of enzymes, and even in the same glycosylation site of the same kind of molecule, the structure of the carbohydrate chain is also different. In addition, the interaction between glycans and proteins is accomplished through the multivalent and intensifying affinity between them, and this force is relatively weak. These characteristics place very high demands on analytical techniques. Therefore, in order to carry out research on the structure and function of glycans, researchers have established a new research method, glycan microarray. Glycan microarrays, first described in 2002, provide a high-throughput means of profiling the interactions of glycan-binding proteins with their ligands. This method can be used to identify specific binding of glycans by lectins, antibodies, toxins, viruses, and so on. In glycan microarrays, individual glycans from natural sources or chemical or enzymatic syntheses are modified to allow themselves covalently or non-covalently attached to a solid surface. The glycan microarray can be interrogated with glycan-binding proteins or other reagents, or even cells and viruses. Through this way, those recognized glycan spots are identified, which can be visualized by either direct or indirect fluorescent tagging. The results indicate that the glycan-binding proteins bound strongly to one glycan, less strongly to another glycan, and do not bind appreciably to any other glycan. Such microarrays can also be prepared from glycolipids, glycopeptides, whole glycoproteins, or polysaccharides. There are many applications for glycan microarray. First, rapid profiling of glycan-binding properties of proteins, including plant and animal lectins, antibodies, and growth factors. To achieve the goals, carbohydrate microarrays are incubated with fluorophore-labeled glycan-binding proteins, fluorophore-labeled secondary reagents that interact with the glycan-binding proteins, or a tag, like biotin or his tag, conjugated to the glycan-binding proteins. And then a high-resolution fluorescence microarray scanner is applied to quantify the fluorescence intensity of the bound proteins. In addition, surface plasmon resonance imaging and mass spectrometry techniques are employed as label-free detection methods in glycan microarrays. In addition, examine glycan-binding properties of whole pathogens and to detect pathogens as part of diagnoses. Many pathogenic bacteria display the pathogenic properties, which are caused by the binding of pathogens to host cell surface glycans through specific glycan-protein interactions. The researchers incubated a microarray containing glycans with the bacteria strain, which was stained with the fluorescent dye. The microspots containing mannose epitopes exhibited fluorescence because of the mannose binding protein on pili expressed by the strain. In order to make this approach become widespread, experimental conditions employed for this purpose should be optimized. Determine the quantitative binding affinities of proteins to glycans. Researchers apply glycan microarrays to determine concentrations of soluble inhibitors to inhibit 50% of protein binding to glycans. In this assay, a series of pre-incubated mixtures of the fluorophore-labeled lectin and an inhibitor were applied to glycan microarrays. The values of the inhibitors were determined by measuring the fluorescence intensities of bound proteins that remain on the microarrays after washing. Employ the microarray technology for rapid screening of functional glycans that elicit cellular responses by interacting with cell surface lectins. Carbohydrate microarrays are constructed by attaching unmodified glycans to the hydrazide-coated surface. Mammalian cells expressing lectins were pretreated with a fluorescent probe, PF1. PF1 can selectively detect reactive oxygen species. The binding of glycan ligands to lectins on cell surfaces can trigger a cellular response that results in the generation of ROS. Therefore, the resulting fluorescence signal emanating from PF1 is directly related to glycan binding to cell surface lectins. And the microarrays containing glycans were incubated with these mammalian cells. The functions were identified by measuring the fluorescence intensity of each spot. The results show that cells adhering to glycans on the microarrays generate ROS, as reflected in an increase in fluorescence signals induced by the ROS probe. As one of the leading companies in the omics field with over years of experience in omics study, Creative Proteomics provides various types of glycan-related microarray assays, including lectin microarray, glycogene microarray, glycan microarray, microbial glycan microarray, and glycopeptide microarray. Thanks for watching our video. At Creative Proteomics, we provide the most reliable services. If you have any questions or specific requirements, please do not hesitate to contact us. We are very glad to cooperate with you.